Thank you for coming. I have to thank Igor Almeida and Doug Watts, who approached Osegeda with a request for $120,000, which brought us this new piece of equipment. And I'm going to leave it over to Will Hauser from PerkinElmer, who just installed it this morning, to tell us a little bit about it. Okay, so
thanks, Dr.
All yours.
Hi, my name's Will. I am an electrical engineer, but if you want to beat me up with molecular biology questions, feel free to do so. Today we're going to talk about the science behind in vivo imaging. What will make your experiment the most successful, the basic physics and biology that will go into what you need for study design in order for your grant to be a success or your research or your abstract to look good. We're going to do a review of the hardware so you understand the components there. We'll do a review of the software so you understand how to use it if you're going to be acquiring your own images, or if your students are going to be acquiring images, how at least you'll be able to look at their data sets and understand whether or not they're on the right track or not. I'll do a review of the limitations involved with fluorescence, because that can be quite challenging sometimes. And then we've already done some hands-on training today. The system is great for qualitative analysis, and this is why oncologists have historically loved this instrument. The more cells you have, the more molecules you have, the more light they're giving off, and the easier it is to see that infection or that cancer inside that cell line. And then if it begins to respond to your treatment, that light will shrink down in size, and then you'll also be able to see whether or not your therapy is effective or not. It's non-invasive therapy, so you actually see the living cells inside the mouse, and luciferase is an amazing death reporter. So as soon as that light goes out, then you know that your cells have died, your therapy is hopefully effective. And it's very, very, very easy to use. So you don't have to appoint an operator for this system. All of the researchers using it can just do all of their own imaging. It's very, very, very simple from that point of view. Most people doing bioluminescent imaging are using bioluminescence, and they're taking a naturally occurring luciferase, such as firefly, click beetle, Gaussia, or Ranilla, and they're transfecting those onto the cell lines that they're studying. Then, in the presence of a substrate, typically D-luciferin potassium salt in the case of click beetle or firefly, or something called solentrazine, C-O-E-L, in the case of Gaussia or Ranilla, if the mouse is alive and breathing, then those molecules will give off energy at the form of light where we can then detect them. Does that make sense? So most people doing bioluminescence have already transfected a plasmid onto the cell line that they're looking to study. There's, I think, 15,000 publications out there in oncology where people have used an IVIS in their research. So it's the de facto standard, and it's what you're probably going to want to use for an oncology grant application. There's also applications out there in infectious disease. Here we can see an ex vivo brain for a neurology application, inflammation markers, and that's probably what your T. cruzi will look like if you're studying that. This is a publication. No, it wasn't a publication. This is data that came out of a pharmaceutical company in the Midwest when they were trying to justify the purchase of an IVIS to their managers. They said, hey, we have to wait about a month before our tumors are sufficiently large enough where we can physically palpate them and put a set of calipers on and quantify their size. However, if we would stick with bioluminescent imaging and get one of these nice IVIS imagers, we'd be able to detect much sooner. How much sooner? Well, if you mix your bioluminescent cells with a bit of substrate, you can see them right at the time of administration. And then basically, as soon as there's implantation and vasculature, you can detect those cells in the presence of a substrate. So you see this nice linear graph in the pink compared with they might have seen something about a month in trying to image with a different modality. So what that tells me is that you guys can now scoop your colleagues. You can publish a lot sooner, and you don't have to wait nearly as long before you begin your treatment regimens, which means you can generate more data in less amount of time. Anybody here from Texas? Right? So you can do plating. This is a bladder infection that's traveling up into the kidney. Here we see bone marrow infections, and the light will go right through bone. The light will go right through muscle tissue. The light will go through anything else that you happen to be working with. The limitation is oxyhemoglobin, and I'll explain that here in a minute. But basically, the further we redshift the color of the emission, the deeper that light's going to penetrate through. So if I have something that emits blue, it's only going to be visible at about 1 millimeter of depth. But if I have something that emits red, it'll be visible at almost unlimited depth inside of a mouse. here's a couple of common bioluminescent reporters. In the blue, I have blue bacteria, and in the purple, I have Renilla. Only the red portion of their emission is significantly red enough to see at any depth. So only that little area right there is actually red enough to see at about 1 millimeter. But if I go with North American firefly, like a PGL 4.5 luciferase, at mammalian temperatures, it's not that green lightning bug that you'll see outside in Houston on a dewy morning. It's actually going to emit orange or orange-red at 37 or 39 C at a mouse temperature. And because of that redshift in firefly, if you go with a firefly luciferase, you'll be able to see a smaller number of molecules at greater depth by going with a firefly luciferase. The obstacle here is oxyhemoglobin. Oxyhemoglobin attenuates light that's shorter than 600 nanometers in wavelength. So I've added a few other common luciferases to this chart. Here I have blue bacteria, and basically I have no emission from blue bacteria or from the standard Renilla. CB is click beetle, and click beetle green happens to overlap with these two peaks in oxyhemoglobin attenuation, so it's not an ideal reporter to go with. Firefly overlaps almost perfectly with CB or click beetle red. So just because something says red or green or whatever in the name, it's not necessarily true. Actually look at the emission of whatever plasmid you're planning on using or whatever fluorescent protein or monomer you're planning on using and see what the emission is at 37 C. If I take my laser pointer and I shine it through a tub of pure water, that light cuts right through. But if I add a few drops of milk to that, now I'm trying to scatter light photons in a turbid media. The more milk I add to that media, the more turbid it becomes, and the more scattering and diffusion I'll experience. To show you that a different way, you're also going to see the same thing inside your mouse. It's not a laser and milk, but there's a bioluminescent source that's not exactly cutting right through like a laser. It's scattering and diffusing, and those photons are eventually making their way up to the surface or the skin of the subject, where the CCD camera up above can look down upon that surface and see the photons being emitted from inside. So position your subjects accordingly. If the source is deep down inside, then you don't have to worry about this. But if the source is shallow, you want to position this subject dorsal so the CCD can look down at this very small number of molecules and see what's right there. If I have a very small number of molecules in the peritoneal cavity or in the gut, I might want to consider flipping this model over and imaging ventral or belly up so the source inside is closest to that CCD camera. This is your system. It's custom designed for in vivo imaging. It has a nice sensitivity range from 500 up to 840 nanometers. So just because we can detect it doesn't mean you'll be able to see that at depth. But the system does have filters, and the CCD does have capability to see just about anything within that range. The system has a nice dynamic range. So Dr. Francia and I are going outside today, and I'm an antique camera buff. And I have all these antique 35 millimeter film type cameras. And I want to take his picture with my film type camera outside right now. It's a nice, bright, and sunny day, right? So we go outside. There's that one star that's shining up in the sky. I take his picture. Click, right? Really quick exposure because there's a lot of light outside. Now, if I wait for the sun to set, in order to take his picture again, I'd either have to use a flash, but if I don't have a flash, I'm going to have to take a really long exposure because there's not nearly as much light out there. But there's a bunch of other stars in the sky. When the sun is out, that's our only star that we can see. But when the sun sets, we can see all the other stars in the sky because our eye has approximately the same dynamic range, about 5 logs of delta. Inside your subject or inside your source, if you've got a large primary tumor or a large infection, if that tumor is so large or that infection is so large, it will be so bright that you won't be able to see the micromets or the other colonies that are adjacent to it without resection, right? But we can continue to adjust the exposure settings and try to detect it using this system. And I'll go into that here in a little bit. If we cut the machine in half, there's an electronics tray, there's a heated sample stage. It helps keep my subjects nice and comfortable for long periods of imaging. There's LED illuminators because it's a light-tight black box. You can keep the lights on in the room while you're doing your imaging. but these little LED flash bulbs will illuminate a subject that's down on the stage. And then up on top, there's a Peltier-cooled CCD camera. That camera will cool down in about 10 minutes so that you can come in, initialize the system, and even if you've lost power, it'll be ready to image, whereas I don't know how long some of the other systems take to cool off, but I know our old ones maybe took 24 hours to cool off. This is much better than that. The Living Image software completely automates the system. So it'll automatically make your stage go up and down. It's automatically going to spin filters into place for you. It's automatically going to adjust the settings for you. And it's also going to provide you the tools that you'll need after you've acquired a bunch of images to go back through all those images, catalog and browse through all those images, and do analysis for your study or for your publication. The instrument settings are analogous to manual photography. So again, if there's a lot of light outside, I'm going to take a really quick exposure, but after the sun sets, I'm going to have to take a much longer exposure. Images are acquired in a two-step process, and here's that process. First, we take a photograph of our subject sitting on the stage, and then we turn off the lights, and we allow the bioluminescent or fluorescent photons to emit enough signal where they can be detected by the camera. Then the software automatically overlays the bioluminescence on top of the photograph so you have a spatial representation about where your light intensity was originating. This instrument has 4 fields of view, actually 5 fields of view. The fields of view range from D, where you get image 3 mice across, to C, where you get image 2 mice across, to B, where you get image about 2/3 of a mouse, and field of view A, where you get image 1/3 of one mouse. If I'm standing way back here, you can see my entire body. But as I get closer and closer and closer to you, then you can look at me and say, hey, that Will, right? I can see him up close and personal. He's a really good-looking guy. If you've got a micromet in the brain of a mouse, you might not see it if it's way, 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 way down here. But if you raise the stage up to field of view A, you stand a very good chance of early detection or late detection, and you'll see a lot more anatomical reference points. So if you're wondering, why are my T cells trafficking to the left hemisphere of the brain and not the right hemisphere of the brain, you can stand your best chance of seeing anatomical and early detection at field of view A, until your biostatistician says you need N of 100 mice for statistical significance. Yes. At which point you need to churn them out as many as possible. So you're going to be down here imaging 3 across, or down even 1 more, imaging 5 across to try and churn them out. So what else can you do? Well, you can increase the exposure time and allow more photons to integrate over a longer period of time. You can adjust the aperture or f-stop and allow more photons to reach that CCD camera, or you can adjust something called pixel binning, and I'll go into these terms here in a second. This is an illustration of my little plastic phantom mouse, and at field of view A, B, C, and D. We have an additional field of view. I think that answers your question. There's a clip-in lens option that was purchased that'll give you a 23 by 24 centimeter field of view E. So for those high throughput studies, you now have this as an option available to you. There's not nearly the resolution. There's a quarter of the resolution you might have otherwise. But if you need that high throughput study and your sources are nice and bright, this is an amazing option that you can take advantage of. The CCD camera has a raw signal range from 0 to 65, 535, or 2 to the 16th. Every day when you come in to acquire your images, you or the auto exposure algorithm will want to adjust the exposure time, the binning, and the f-stop or aperture in order to raise your count above 600 and lower your count below 60,000. The reason that you or the auto exposure algorithm is able to change the exposure time, the binning, and the f-stop is because this instrument's calibrated. So it's able to normalize between a 1-second image and a 5-minute image. It's able to normalize between one day and another, and it's calibrated so you can co-collaborate on that secret grant, and you can call the guys in Houston that have an IVIS, right? And now you guys have an IVIS, and you can co-collaborate with anybody else out there that has an IVIS and publish together if you want. This is the control panel and where you would adjust for exposure time, binning, f-stop, and the field of view. Probably the most intuitive thing to adjust is exposure time because it results in a linear quadratic change in the amount of counts being detected. In these two examples, I went from 2 seconds, holding my f-stop constant at f1 and my binning constant, small binning, up to 10 seconds, again f1 and small binning, and I went from 5,000 counts quadratically up to 25,000 counts. So you guys can do the math in your head. 2 times 5 is 10, 5 times 5 is 25. How adjusting that will result in a change. The shortest exposure time that we recommend with this system is a half a second. Anything shorter than half a second, and the infection's probably sufficiently large, at least on a mouse, where it might be time for euthanasia on that subject. Anything longer than 5 minutes, and there's most likely an issue at play with your biology. I know plenty of people that are able to see 3 cells, bioluminescent cells, at about a minute of exposure time. So if you're not seeing signal at 5 minutes, feel free to call me, but we might want to take a look back at your methodology and your study design and just make sure that the transfection went well and along the way when we looked at those cells in vitro or in, prior to in vivo, that they were nice and bright and stable cell lines. You can also adjust binning. The binning settings are large, medium, and small. Holding my exposure time on my f-stop constant, at large binning, I have a very pixelated image, but the counts are quite high. The counts are at 32,000. At medium binning, it looks a lot less pixelated, but now I'm down to 8,000 counts. And at small binning, I have a lot more resolution, but now I'm way down here. I have 1/16 of that resolution, and I'm only at 2,000 counts. When you go to apply for the grant, is it an anatomical focus on the grant? When you're going to graduate, is it necessary that you have an anatomical small binned image that's a single well on a well plate that's blown up on a 22-foot projector screen? Or are you doing a screening study? Are you putting 100 mice in for 28 weeks and seeing which of those 100 mice are responding to your treatment regimens? If it's the latter, then you can save yourself quite a bit of time by going with large binning. I could take a 15-second image at large binning, whereas I would have to take the exact same subject for 4 minutes at small binning. So you can save yourself quite a bit of time and save the mice quite a bit of time under anesthesia if you don't have to, and it's not necessary for your publication to get that level of resolution. Does that make sense? Okay. You can also adjust aperture or f-stop. The f-stop settings range between 1 and 16. At f1, the aperture is almost all the way open, allowing the most amount of light to get through and hit the CCD camera. So this is what we're going to use by default for bioluminescent imaging. At f2, you'll have a quarter of that sensitivity, and that's what we default use for our fluorescent imaging. And then all the way down to f16 is what we use for our photographic imaging, because it brings everything on the field of view into perfect flat focus. We typically don't use aperture as a tool for adjusting focus. Or we have this really nice feature called auto exposure. Now I wanted everybody to understand the basic behind exposure time, f-stop, and binning. But now, who here has a cell phone in their pocket? Who here uses their cell phone to take pictures? Everybody? All right. Who here uses the exposure time, f-stop, and binning on their cell phone when they take pictures? Why not?
Do we have that?
Yeah, right, because auto exposure works just fine, like 99% of the time. So does this. So in order to get to auto exposure, over here where it says exposure time, start clicking down. I think it says 1 second by default. Click it down to 0 seconds, and then one more click down will bring up auto exposure. Auto exposure is not only going to adjust exposure time, but also f-stop, and this should say binning. It's also going to adjust binning for you in order to hit a target that you specified. The only limitation, or the only reason why you might not want to consider using auto exposure, it has a maximum exposure time of 60 seconds. So anybody working on adaptive immunotherapies, anybody working on Chagas that has like fewer than 1,000 T. cruzi in the mouse, anybody working with, micromeds, along those lines, auto exposure might not afford you the sensitivity that you need in less than a minute of exposure time in order to see that. But for anybody working with large infections or large solid tumors that are giving off a lot of light, this is going to save you quite a bit of time and take all the worry of having to worry about exposure time, f-stop, and binning away from you. Just to review the control panel, exposure time, binning, f-stop, and the field of view. In order to take a single image click, and a single image consists of a luminescence being overlaid on top of a photograph, or a fluorescent being overlaid on top of a photograph, click the big button that says Acquire. In order to acquire more than one image, click this button here that says Sequence Setup, and that window will open up. Now, why might you want to acquire more than one image? So I haven't worked with this strain of T. cruzi before, and I haven't put the T. cruzi into this particular mouse model ever. So I know that as I inject the substrate into the mouse, at a certain time after I inject the substrate, there's going to be a pharmacokinetic, there's going to be an emission, bioluminescent, and then that's when I want to do my imaging. But when is it? I read all these papers and everybody just picks arbitrary numbers, but I'm going to teach you guys the right way to do it. The right way is just take one little feeder mouse, right? Not the forever mouse, but one that's positive. Make sure he's positive, inject the substrate, and then begin to image that mouse. Every minute, on the minute, for the next half hour perhaps. And then you'll see the emission from the T. cruzi increases, peaks or plateaus, and then decreases. Now you know, all right, for my infection model and my mouse model and my strain of T. cruzi, I want to do all my imaging, this is an arbitrary number, at 18 minutes after substrate administration. Whereas for the micromets that he's using that are on the kidneys, his peak might be 6 minutes after substrate administration. But you know that from this point forward, your techs are always going to be doing the imaging at 18 minutes after substrate administration. So they're always going to be comparing apples to apples to apples, and they only have to do this one time. And then your students looking at those mets on the kidneys will always be imaging at 6 minutes after substrate administration, and then all of their images won't be dependent on the luciferase pharmacokinetics. Does that make sense to everybody? In order to do that example, I would check this box that said number of segments and make that about 30 with a time to live 1 minute. And then over here is auto exposure, so when I say add, it's going to add an auto exposure that for every minute on the minute for the next 30 minutes, it's going to acquire an image of my subject sitting there on the stage. Does that make sense? Okay. We have this really nice button here called the Imaging Wizard. This is going to ask you a series of point-blank questions, assuming that you don't start imaging for a couple of years and you've forgotten everything from today's training. Don't worry, the Imaging Wizard is here to help you. The Imaging Wizard asks you, are you doing bioluminescent imaging or are you doing fluorescent imaging? I'm doing fluorescent imaging. All right, what's the agent that you're using? Oh, I'm using this ProSense 750. Okay, well, we know that depending on the depth of what the host would be inside the subject, that these 4 excitation filters are going to be the best, and this 1 emission filter is going to be the best. Why don't you start off with these and see which one of those 4 is best for your ProSense down in the liver of your model. So this is a really nice way to familiarize yourself with the system if you're in a real hurry and you don't want to read the manual and figure out how to do it. As you acquire images, you'll see this pop-up window open up. You can fill in up to 15 of these metadata fields with information for you to find your images at a later point. If you want, you can put a checkbox next to them, and the images will be pasted up at the top of the screen. But that's not the really important part. The really important part is save all of your images in one folder. For one experiment, all of your images just in one folder. Don't attempt to organize the experimental folders. Don't attempt to organize the images sets. Just save everything for one particular experiment in one folder. However, do an effective job labeling the metadata fields. And then when you go to look at your images at the end of your experiment, go to File and then Browse. And just point it at that one folder for your one experiment where all of your image sets are saved. Then you can sort. You can sort via multiple columns here so that you can find your images from day 7 with this treatment group and day 7 from this treatment group, which might not have been on the exact same dates. They might have been months apart, but you can compare day 7 to day 7 to day 7 to day 7 and actually see what's there. If you're not correlating anything, then you can track them longitudinally. And you can say, okay, I want to compare time 0 to day 1 to day 7 to day 14 to day 21 to day 28. Load as a group, and it's going to load all of those images side by side by side, which makes your life a lot easier, and it'll make your publication go a lot easier because now there's only one minimum and maximum scale that you just set all of your images to simultaneously. You only have to draw one set of regions of interest, or ROIs, and they're automatically pasted on each of all the subsequent images. And it's all done for you, and then you can just export that image as a TIFF, drop it into your abstract, and continue on with your life. So, yes?
So initially you're looking at something called DS Sense, and now you're looking at DS Red. And I guess my question has to do with how sensitive, because I don't know if you have to use the dyes that you're talking about, which were in the page 3 or 4 before this. Earlier you were talking about luciferase vanilla, and now you're talking about DS Red. So
I haven't even gotten to fluorescent yet. This is just an example, but yeah. So you want to know what the lower limit of detection is?
I'm just trying to figure out what, when you're showing these images, because you're going.
So everything that we've spoken about so far has been bioluminescent. The lowest limit of detection with bioluminescence is basically 1 cell if you've done your homework. Now, 10 years ago, I might have said 3,000 cells at about a centimeter of depth, but most of the luciferases have gotten more and more codon optimized. People's transfections have gotten a lot more improved, and there's different ways of sorting now so that you can ensure you have 100% efficacy on everything that you're injecting into that subject is bioluminescent.
And that sensitivity is based on a well-vascularized region, because you have to be able to get.
Yeah, so necrotic tumors are a whole different bag. Yeah.
For the brain.
That's not an issue at all. Like,
so the substrate goes across to the brain.
Red laser dot. You guys can see the red laser dot is going right through the bone in my finger.
Right. No, I'm thinking the blood-brain barrier. So does the dye go through does the substrate go through? Substrate
is D-luciferin potassium salt, so potassium will cross BBB. Dyes are a whole different bag that we haven't touched on yet.
No.
Yeah.
I'm trying to understand what you're saying in the context of what I'm interested in.
Yeah, what are you interested in?
So we're interested in looking at cells moving in across the blood-brain barrier.
What kind of cells?
T cells.
Oh, yeah. All day and all night.
Okay.
Yeah. Yep. No problem. Okay, so back to this though. If I've saved all my images in one folder, then I can go to File, Browse, load as a group, select the different images from the different time points, and put them all together. Put them all together, do analysis. Now this is a single image that I'm going to do analysis on. My very first step is to go in the upper left-hand corner and go from the uncalibrated relative light units of counts to the calibrated units of radiance or photons. And then it won't matter what my exposure time was, what my field of view was, or what the distance of my subject to the CCD was. Everything's completely calibrated and completely normalized. Then I can draw a region of interest, or ROI, around the area that I want to study. The ROI shapes that I can draw are a circle, a square, a grid if I'm doing plate work, or a contour. This button will measure them, and this button will delete an ROI that I don't like. I can also set a minimum and maximum. So if I want to focus on the micromets, and they're just little hills next to the big primary tumor mountain, I can actually lower the max down so that people can kind of look at my publication and really see and focus on the mets. Conversely, maybe I'm not interested in treating or not funded for treating the mets and I'm only funded for treating the primary. I can raise the minimum bar up and eliminate any of that signal, at least the visual portion of it, from this particular view. It'll still be visible when I quantify radiance, however. Here the ROIs are being drawn. A square, which I like to make into a big rectangle and encompass the entire subject. And then I'll see an overall metastatic burden. I'll see an overall amount of infection. I'll see if there's cruzi outside of the pause, and maybe I do have cruzi inside the liver. If there's a particular area that I want to focus on, I can draw a circular ROI around the area. I can allow the software to set a contour or border around the area, and I can also do plate work using a black, clear bottom polystyrene imaging well plate. I can bring those cells or molecules down in front of the machine and draw a grid ROI around them for analysis. After I've drawn the ROI, this button will measure the amount of signal that I have here. When I click the measure button, you'll see this pop up. Now, most of the publications that I'm looking at, people are imaging units in total flux, or photons per second, and they're comparing that versus date and time for longitudinal, for your X and Y axes for a longitudinal analysis. When you click the measure button, if you don't see the values that you're looking for, or in that earlier pop-up window where we were filling in the metadata, if you want to write a comment, this is mouse number 3, this is mouse number 4, and this is mouse number 5, so you can have a yellow line, a red line, and a blue line to track out 3, 4, and 5. You can configure this view to include the mouse number or whatever else you feel like graphing out that'll make your life or your publication or your grant application go a lot easier. This is that example from before where I went from 2 seconds and 5,000 counts up to 10 seconds and 25,000 counts. But instead of looking at my images in the uncalibrated relative light units of counts, I want to look at it in the calibrated units of photons. Total flux, photons per second. I have the same light source putting out the exact same amount of light, so I'm completely calibrated and normalized. Let me show you a different way. I'm using auto exposure here. On day 1 of my experiment, auto exposure came in and took a 30-second image at small bitting on day 1, which registered at 1,600 counts. On day 2, auto exposure took a 30-second image at small bitting on day 2, but now it was down to 800 counts. Following the slope of this line down, I can then see on day 3, I would have fallen well below the 600 counts that I want to stay above. So auto exposure increased my exposure time from 30 seconds up to the maximum of 60 seconds, which worked fine on day 3 and fine on day 4. But on day 4, I'm approaching that limit of 600 counts, and I want to stay above there. I can't increase, or auto exposure can't increase the exposure time beyond 60 seconds. So instead, it increased my binning, going from small up to medium binning, which raised my counts back up. Now, if I show you the series of images, and I show you this graph and say, hey, there's a tumor in the left leg of my subject, it's really responding really well to my treatment, everybody has to scratch their head and say, what in the world are you talking about? I don't understand. Instead, if I show it to you in the calibrated units of radians, it normalizes numerically, it normalizes my image visually, so you can show anybody exactly what was going on. Of special interest, do you see how that little area right there is this water-filled necrotic tumor, but only a very small portion of it is bioluminescing? If I were really trying to see this with any other modality other than bioluminescence, I probably wouldn't be able to see what's there. But by using bioluminescence, I can see that only a very small portion of it continues to bioluminesce. So bioluminescence is a great death reporter, because as soon as you no longer have oxygen or ATP, or as soon as your cells are dead, they no longer bioluminesce. This is my buddy Brian. Brian used to be at MD Anderson 10 years ago when he published this in PNAS. He's since gone on to fame and fortune. These are murine T cells. Here we have 3 murine T cells. Murine cells are cryptic splice sites. They're a pain in the neck to transfect. 10 murine cells and 30 murine T cells. What Brian did was come in, mix the cells with a little bit of substrate, did a sub-Q injection, saw them fire up right away. This is a 1-minute exposure time. And then he published a methodology paper. He sent it off to Major Methods. They didn't want it, and he went into PNAS back when it didn't even have a good impact rating. But this isn't to give you any type of false hope. It's to encourage you to do your homework. This didn't just take Brian a minute. It took him a month. It took him a month to codon optimize, codon by codon, a PGL luciferase. And then, back then, he didn't have a really good linker. He had to use IRES as his linker. He linked his plasmid via IRES to an EGFP because the only cell sorter he had at the time had a blue laser on it. So he could sort for GFP. He couldn't sort for luciferase, but by sorting for GFP, everything that was GFP positive was also Luc positive. So he was able to then sort out and get 100% of the cells that he knew were bioluminescent out of there. And he found a really elegant vector that worked, helped to overcome those cryptic splice sites that murine cells have. So this was the end result. I've met plenty of people who are struggling, to post-process cells that were labeled with GFP, right? Your cell line might have GFP on there, but just add a linker of some luciferase, at which point you'll be able to use the GFP for all your ex vivo and all your in vitro stuff, and you'll be able to use the luciferase for all your in vivo imaging and see really nice results. Any questions about this one? All right. Anybody planning on doing fluorescence? Okay. So we can do fluorescence. The way fluorescence typically works in a microscope is we're going to excite a fluorophore at a particular wavelength. We're going to drive that excitation light in. There's an electron inside the fluorophore. Once, much like yourselves right now, you can't contain your excitement anymore. It decays and emits light at an upshifted wavelength. Well, that's how it works in a microscope. In a mouse, we try to drive the excitation light in, but it hits the black fur, and 92% of it's attenuated. Oh, geez, what do we do? So we shave the mouse, but it still has black down in the follicles. So then we put Nair, or a depilatory cream, on the mouse. Okay, so now it's just raw skin. So now we can shine the excitation light at the raw skin, at which point all the proteins in the skin autofluoresce, and all we're seeing is autofluorescence. All right, so we can filter some of that out, and now we're driving the photons of excitation light in, and then they hit myoglobin. And if they're not red, they're attenuated out. And then they hit hemoglobin. And if they're not red, they're attenuated out. And then they hit bone, and they refract a bit. And eventually, 1 out of every million or so photons hits the fluorophore that we were looking for. And it takes the exact same path on the way out. It hits bone, it refracts. It hits heme, and it's filtered out. It hits myoglobin, and it's filtered out. It hits the skin, and it causes autofluorescence. And, oh, wait, this one went out the side. And remember, our camera's on top, so we didn't catch that one either. But overall, fluorescence is a much noisier process than bioluminescence. Generally speaking, when you're taking bioluminescent imaging, the only thing giving off light inside that machine is your bioluminescent cells, your bioluminescent molecules. When you're doing fluorescence, everything is autofluorescing, and it's a real challenge to overcome that. There's additional challenges also to bear in mind. Additional challenges being oxyhemoglobin. Anything that's shorter than 600 nanometers in wavelength is going to be completely attenuated out by heme. So GFP is not an option, basically, for anything that's beyond the skin. DS red happens to overlap with these two peaks in oxyhemoglobin attenuation, right here and here. So DS red uses green excitation light. Don't ask me why it's called DS red. This is also where M cherry and TD tomato, if you're interested in a monomeric or a dimeric approach, happen to fall. So there's a real challenge to see anything in this wavelength at any significant depth, because it's challenging to drive green light through tissue. A better approach would be something in the Psi 5.5 wavelength or the Psi 7 wavelength. ICG is indocyanine green. Even though it's near infrared, it's one of the better reporters you can go with. You can go with Psi 7 or Alexafluor 750, DIR or IR 800CW. Here, Alexafluor 680 or Psi 5.5. Our fluorescence system has a 150 watt tungsten halogen light source. There's a fiber optic cable in the back. There's 19 excitation filters. That excitation light is going to come down. It's going to cover the entire stage. The emission light is going to come up. Your particular machine has 4 emission filters configured on it. These are your excitation filters. So we can hit just about anything we want to work with. But in all reality, you probably want to work over here in this region, because otherwise you're not going to see whatever you're looking for at any depth. Unfortunately, you only have 4 filters on your particular machine. They're a little bit different. But in order to take a fluorescent image, you're going to check the box for fluorescent. You're going to choose an excitation and emission filter. Choose a lamp level of high if those cells are inside of a mouse. A lamp level of low to prevent photo bleaching if your cells are in vitro and you want to snap them for a couple of scans. However, don't ever take cells that you've scanned with this system and then plan on put them inside a mouse, because they'll be completely photo bleached out. We operate in calibrated units when it comes to doing our fluorescent imaging. Those units are photons per second per square centimeter per radian over microwatts per square centimeter, otherwise known as radiant efficiency. But what that actually means is that we compensate for any excitation light pattern that might be down on the stage. That excitation light pattern concentrates in the center of the field of view due to barrel distortion, due to vignetting. We just happen to see more signal in the middle. So if I had a well plate, and every single well in this well plate contains the same number of cells, but I try to look at it in counts, it appears that the center wells of the well plate contain more signal than the wells off the edges. However, if I switch to the calibrated units of radiant efficiency, it normalizes for this excitation light pattern, and I can see that indeed I did have the same number of cells in every single well of that well plate. These are control mice. These are negatives. At GFP wavelengths, mouse tissue autofluoresces like the dickens. So you can't drive the blue light in, you can't see the green light coming out, and on a sham mouse, the autofluorescence actually has about the same quantum yield as the GFP itself. So GFP is not really a good option. Now this is where DS Red, M Cherry, and TD Tomato happen to fall, and unfortunately there's quite a bit of autofluorescence to contend with. So we're trying to excite this using a green light source. It's really, really, really difficult to drive that in, and if we were able to get it in, then on the emission side we still have to contend with all this autofluorescence. At Psi 5.5 and Psi 7, at Alexa Fluor 680 and Alexa Fluor 750, at ICG, DIR, and IRCW, there's almost no tissue autofluorescence to contend with. So that's one more reason why I'd like to recommend that if you are still intending on doing fluorescence after me mentioning all the negatives involved with fluorescent imaging, that you stick with something at the 680 and 750 wavelengths, because otherwise you're going to have to spend a lot of time trying to subtract this out and differentiate this autofluorescent signal from the real signal, which, here's one more limitation. If you're planning on doing fluorescent imaging, you want to transition your subject off of a diet that contains chlorophyll at least 14 and ideally 21 days prior to beginning your study. With bioluminescence, this is no longer an issue. Once you transition them off of regular rhododendron alfalfa-free, 2 to 3 weeks later, you'll see far less autofluorescence in the gut of the subject. He is the principal investigator of the Giardia. Giardia? All right. Okay, so for your Giardia, we might want to consider switching them to an alfalfa-free. I know it's a bioluminescent Giardia, but there is a little bit of emission in the rodent diet that accumulates just from the ambient lights in the vivarium coming down, and it'll charge up the way an exit sign. Have you ever seen those phosphorescent exit signs? When you turn off the lights, they give off that green light. The chlorophyll from the alfalfa will charge up in the room lights, and you might see a bit of signal. If you're trying to look for only a few thousand Giardia that are in the gut, I wouldn't want to confuse the signal from the Giardia with the signal from anything else. So your other option, if you don't want to go with an alfalfa-free rodent diet approach, is just to put a few mice on the stage. The first mouse is a naive, a negative, who's on a regular rodent diet. The second one can either be Giardia positive, and your third one's Giardia positive as a treatment group. So you have 3 mice there on the stage. You can use the signal out of mouse number 1 as a baseline and say, okay, even if there were nothing in the gut, this is what I'm going to see, 1 times 10 to the 4th, whereas your Giardia is 1 times 10 to the 6th, and your treatment's 1 times 10 to the 5th. So you know that where baseline happens to occur at. Otherwise, you might want to transition them off of regular rodent diet about 3 weeks prior to beginning the study. It has a metabolic effect, however. We can subtract off for tissue autofluorescence. This is probably not going to enable you to see GFP at depth. It's a very time-consuming process. There's about 15 pages in the manual that explain exactly what you can do here. But if you happen to notice quite a great deal of tissue autofluorescence and you're still insistent upon doing fluorescence imaging, you can subtract off for any of that autofluorescent signal that's there. Yeah, that's fine. Choose a reporter that's going to give you the best signal-to-noise ratio. In my opinion as an electrical engineer is bioluminescence. I understand that for your biology and your methodology, sometimes the only option is fluorescence. And as long as you understand that and you've chosen something that's red or near infrared, ideally, then that'll be best for you. Consider the appropriate amount of control group. So if she's seeing all of the signal from the gut, right, then by adding a negative to every single set of 3 or every single set of 5, she'll then kind of know where baseline happens to be occurring. Using a hairless model will be the best. If you can't go with a hairless model, take a white furred model and shave it, or use a depilatory cream like Nair on that. If you're going to be imaging anywhere around the gut doing fluorescence, you want to switch to an alfalfa-free mouse model. Understand the kinetics of your bioluminescent model, and ideally, before your study begins, just do it one time. Do a kinetic study so you know at what time point after substrate administration to do your imaging, so it's at its peak. Animal handling can significantly affect kinetics. So if I inject my subject with substrate and then put it on a wheel, it will no longer have a peak at 10 minutes after substrate administration. It might have a peak at 5 minutes after substrate administration. If he's having a difficult time treating a necrotic tumor model, however, and then he injects his drug and puts the mouse on a wheel, it might have far improved outcomes versus a mouse that's just receiving an infusion of the chemo drug under anesthesia. Change the orientation of your subject, dorsal or ventral, so the cells or the molecules or the infection inside is closest to the CCD that sits on top. Cover up intense signal, so if you have a super bright tumor or a super bright infection, you can wrap that up with black electrical tape. You can wrap that up with aluminum foil, and otherwise try to obfuscate some of that signal that's there if it's more than 5 logs of delta from the most intense to the least intense signal on that same subject. There are little spots in the anesthesia manifold inside the IVIS where you can put guards to segregate light from one subject to the other. And then if you're going to be doing any type of plate work, use the black walled, clear bottom polystyrene black imaging well plate. There's a very nice manual. There's a link to it in our software, and there's a link to it on the desktop. That link is no longer correct. Don't record that. We have a full line of probes. You'll notice that most of our probes are in the 680 and 750 wavelengths because that's just what happens to work best. There's activatable stuff. There's stuff that cleaves and then begins to fluoresce. We have a full line of bioluminescent cell lines. We have a whole bunch of lux plasmids in bacteria, and we have injectable substrates and injectable luciferins. That is my Houston cell phone number. That's my email address in case you guys need to get a hold of me. And that is the phone number for technical support and the email address for technical support. This is on the desktop of the IVIS computer down in the vivarium, as well as my business card will be on the side of the IVIS. If you're ever down there and you have an emergency, call me.
Great.
Questions?